Welcome everyone to our sixth session of our 6-month Biocontrol Technical Workshop series. I've already done a bit of an introduction to many of you. We had just had a few technical difficulties at the start with the sharing of the slides, but it looks like it's all fixed now, and thank you for Roma and Pathi for working that out. It's wonderful to be here again. I'm going to move quite quickly to make up a bit of time. So I'm going to move to the next slide. And this is just how to use the system today. You all probably know how to do this. We've had lots of experience throughout the last 6 months. If you have any technical issues, please just try logging off and on. Actually, it's probably the best idea. Or go down to the mute, and if you press the little arrow there, you'll see system settings, and you can click onto that and you can actually look at what your sound system is and also your video selection there. So that will sort out other problems. If you would like to ask a question today, and we really encourage that, please write them in the Q&A like normal. It's really helpful for us to have them all in one place. Please use chat though to talk to the group, to share your resources. If you're working on something and you want to tell us what you're looking at in biopesticides and your experience of trials, please share that with us in the chat. We love to have that. And also you can rename yourself if you would like, just by pressing the more button. If you slide over your participant name there. Now, I have a bit of a thunderstorm in the background, so hopefully that won't be too loud. If we move on to the next slide. And this is just a reminder that this is part of a 6-month workshop series. We've actually developed this with a number of different organizations across the world. We're very interested in your feedback, and we now have a website, a portal there for you to communicate with us, and that's at the ASEAN FAWaction.org. So please visit that. If you wish to have a certificate of participation, and I know many of you ask me, you must subscribe to the Biocontrol forum on that website, and you have to either ask a question, share something interesting about yourself, or note something useful in the Biocontrol series. And on the next page, I'm going to show you just how to do that. It's very, very simple. And you'll see here, you go to the website, ASEANFAWaction.org. You then will check on the community link there on the menu, and then you'll click on forum. You'll go to the forum, and you'll see at number 3 there a picture that says the Biocontrol for FAW, and you'll click on that, and it will ask you to probably subscribe. It's very quick and simple, and then you can put in your details, questions, share your research.
Sorry.
No, that's okay. Actually, that's great, Roma. On the next slide, I just wanted to remind you that our next session will be around design tips for conservation biocontrol studies and programs. And then we will also be coming back later in June for another biopesticide efficacy part 3. We will be looking at ASEAN-based researchers and their experiences with biopesticide trials. Okay, next slide.
It's not, sorry, it didn't work.
That's okay.
Nothing's happening.
If you just press, you might have to press it twice, Roma, maybe.
I've done that. It's still not working.
Can you press your down button?
I did that. It's still not working.
That's okay. What we could do is we can just, nothing's working.
I'll try again. Oh, there we are.
Ah, there we go. So I'm going to, sometimes it does get a bit stuck. So don't panic. Roma's probably a bit stressed at the moment because she is going to be presenting soon, and she's also moving the slides, but she's doing a wonderful job. So we actually have her up first, but just to say for this session, there will be some polls. There'll be lots of question time, and there'll be some really good presentations. I'm really looking forward to them. Like usual, our experts have really outdone themselves, and I've had a sneak preview, of course, and there's some really interesting experiences and learnings to share. So I think first up, Roma, you're going to give us an introduction, sort of following on from your session last time, just to remind us why we're all here. And I'd just like to say thank you very much. Your session last time was wonderful. I had such a huge amount of great feedback about it. So please relax, chill out. Don't worry if things don't go right with the slides again. I can always load up another set of slides. So
thank you, Alison. Can I just
welcome.
confirm, are you seeing any of the Zoom operation pages, or are you just seeing the
No,
slides?
I'm just seeing the slides at the moment. It's looking good.
Okay,
Yep.
that's good. Okay, all right, thank you, Alison. And good afternoon, good morning, good evening to everybody who's here, and thank you for joining us again. What I first wanted to do was just go through a little bit of a recap on what we talked about last time, but also I want to draw something slightly different out of it. So just first of all, just a reminder of the technologies that we're talking about when I'm talking about biopesticides. There's 4 groups. You've got the macroorganisms, which are natural enemies, microorganisms, botanicals, and semiochemicals. What I really want to emphasize and to sort of have us thinking about is that these bioprotectants, they have multiple modes of action on the targets and in interaction with the plant, and this makes it more complicated for doing trials. And so what we had looked at last time was what actually happens when we run efficacy trials. And what we could see that if you look at this graph where it's the results and the variability that you get in a trial series, we can see that we get quite a wide data scatter. And the questions we've got is, how do we manage that? What do we do about that wide data scatter? And the data scatter is so wide because you have the variability in your plant, you have the variability in your active substance, and then we have the human variability. And what we're trying to sort out today is that we can reduce the human variability that we put in the trials and see if we can stabilize things a little bit. And what happens when you do trials and you're not managing variability is you end up with data that looks like this. Which if you look at you've got efficacy against the number of pests and looking at the pest population, you have a wide scatter of data. But also this type of, and looking at the data in this way also sort of gives us an indication that I often hear people saying, oh, the product didn't work because the pest population was too high. And when you look at a graph like that, you think, actually, that's not a true statement. What's important is the rate of the population growth, not the size of the population. And you need to sort of just change the thinking a little bit on that. And then if we think about the variance again, what does this look like when we have high levels of variance? So if we look at a product that's doing well and we're getting sort of a good average efficacy of sitting at 70, 80%, but we have a variance which is around about 50 and 60%, what it means is this is what our data will look like. We will have data points sitting within this margin. So it's that slight complication is the better a product performs, the wider the data scatter, and that makes it much harder for us to work with it. And so what I want to try and go through a little bit today is saying, how can we reduce that data scatter? How can we design our trials to reduce that noise so that we can see really what's happening with our product and how our products are performing? So when we're working with biological systems, the biopesticides, they multiply the variance in the system, and it's the variance is the outcome. It's the multiplication of the natural variance in the target population and the plant and the biopesticide population. This is why we have a wide data scatter. So if we can hold that in mind, as I say, it will always be variable. It will always have that effect of the biology. We can't change that. In many ways, we want to harness that. So the trick is, how do we do trials and manage that? And that's what I'll sort of go on to in detail shortly. So first of all, I want to just to see if we're all awake a little bit and just have a little bit of think and just to understand who's in the audience today. So Alison's going to put up a poll now, if you could answer this.
Great, and you should be able to see that. I can see people voting already, Roma. So there's
Good.
3 questions there, everyone. Oh, and voting's happening quite quickly. So we've got 190 people online at the moment, so that's fantastic. And from all around the world, I see all sorts of places. It's great.
So
We've got 902 people who have voted so far, so I'll give a bit more time.
Okay.
Because we've still got quite a few coming on once they sort of slow down. Maybe now I'm going to end polling and share results. Can you see those results?
Well, so we have got, I'd say 50% of the audience has been doing trials with all the technologies, and that, which is really interesting. What
Fantastic.
probably is, it may be that it's not the same 50% have done trials on all three. So I'm guessing that different people have done different trials on different technologies. That's good to know. So this is something that we've all seen the data, we've all seen what the data looks like, and we all have to struggle with trying to understand the data. Thanks, Alison.
So we should have lots of questions, I think, probably, and lots of experiences there to share. Thinking of, that's quite a high number. I'm impressed with that 50%.
Yeah. And are there any urgent questions right now, Alison, that we should answer?
No, not now. I think we could carry on. Yep.
Good. Okay. So we'll go on. I am trying to move on, don't worry. Just a few glitches. Again. There we are. Right. So what's going to happen now is when we're doing this work, we're not talking about the macroorganisms, and I will talk about microorganisms and botanicals in a short while. But what I'd like to do now is to hand over the presentation to Pathi, who is going to talk about chemochemicals and pheromones. Pathi, if you'd like to pick up from here.
Great. And you're okay, Pathi. I'll just give you a quick introduction. Pathi is the Director of Field Development at Provivi and has over 20 years of experience in the crop protection industry and research sector. And he did develop this presentation with Chris Wheeler. So a big thanks to Chris, who I know is in, I think, in maybe San Francisco or somewhere in California. So it's a little bit early in the morning for him. But thank you to both of you. Pathi, welcome.
Thank you. Thank you so much for introduction, and good afternoon, everybody. And today, thanks for the opportunity for speaking about mating disruption, which is a new technology for most of us in Asia. Mating disruption is not new for the world, but it is very new for developing countries, especially for broadacre crops for like rice and corn. So today I'm going to share with you the lessons that we have learned conducting trials in Asia Pacific, especially on rice and corn. Many of the examples that I'm going to take are from corn that we have done trials in Asia Pacific, especially in Southeast Asia. Next slide, please. So let me give a quick introduction about mating disruption. I remember giving a talk on mating disruption or sexual confusion a couple of months ago. But what is mating disruption, or what we call a sexual confusion? This is usually done with insect synthetic sex pheromones. The sex pheromones are naturally occurring sex pheromones where female will give out sex pheromones that olfactory senses will be received by the males to get attracted to and copulate. And then female will produce eggs, viable eggs that will produce larvae. And the larvae will feed on the plants and then go to pupate, and the next generation would occur. So where we intervene with mating disruption is during the starting of the cycle. So the principle behind mating disruption is saturating the whole environment with pheromones, okay, so that the natural pheromone that is released by the females is camouflaged and the males will not find the females. Even with a weaker signal, it's in the huge pheromone cloud, the females will not mate, and the benefits of mating disruption are seen. So I'll talk about benefits of mating disruption in the next few slides. So the whole technology sounds very simple, but there are lots of things that goes on in the background. For example, we know that natural sex pheromone or synthetic versus synthetic blend could be very, very similar. For example, we are mimicking the naturally occurring sex pheromone by synthetically manufacturing them. So when we are doing that, we want to be as close as to the natural sex pheromone, but there are several things that could go wrong. The natural is natural and synthetic is synthetic. So the first thing that we do in the Discovery Science program of characterization is to understand the natural occurring blend as compared to the synthetic blends that we make. So we call these as blend optimization, because The natural sex pheromone does not comprise of a single active ingredient. It's a mixture of active ingredient in specific ratios. So optimizing that blend is the first step towards successful mating disruption program. So it is done both in laboratory as well as field situations. On the left side, you can see that in the laboratory situations, we use electroantenograms or wind tunnels to understand how the males will respond to the synthetic female source. So you see that flight tunnel, what we call as wind tunnel or flight tunnel, where on the one side, we actually put the pheromone source and we release the males on the other side, and we see the upwind flight towards the pheromone source. And we also use electroantenograms to understand specific reactions in the olfactory senses. So once this is all done in the laboratory, we do the field trials. We start the field trials because once we know that this pheromone blend is attractive enough to the naturally occurring pheromone source, we take them and put them in the traps. On the right side, you can see that bucket trap, which was usually seen in maize or corn, where we use for fall armyworm trapping studies or monitoring, for example. So we put these naturally occurring, the synthetic blends of pheromone source along with the natural blends to understand what is the difference between the natural blend and the synthetic blends. And then these are done in representative areas where we want to do field trials to look at the efficacy. So what we call them as trapping studies to understand the attractiveness of the blend. Next slide, please. So once we have the blend, the next thing that we need to understand is where do we put this blend, right? So we all know when we think about pheromones, we all think about lures and traps, right? So even in the lures, we have the same synthetic sex pheromone, but it is in a very small little quantities. It is in milligram or picomolar quantities that is released. In the dispensers that you see on the right side, there's a black things are dispensers, which are made up of a low density polyethylene. It's like imagine a small shampoo sachet. So it has an active ingredient inside, and the materials are very porous so that it will start releasing the pheromone from the dispensers. So these are dispensing the pheromone into the air throughout the season. The formulation itself is a very tricky part because we want to release this pheromone throughout the season for 90 days period without even changing the dispensers. So it's an uphill task. So we want to really understand in the laboratory as well as field situations, how does these dispenser technology actually works. And from Provivi, we also have formulations, which are sprayable formulations. But today's session, I'm going to concentrate more on the point sources or the dispensers. So we really want to understand what happens with these dispensers in the environmental chambers in the laboratory. We want to understand the active ingredient composition. Does it change over time? Is it intact or not? Does it degrade into components because of environmental factors, especially temperature, humidity, wind velocity? Several things can affect the release rates of these pheromone dispensers. And then, of course, the physical stability of these dispensers. It's made up of plastic, polyethylene, so it has to be intact without any delamination through the season. And as we know, the corn plants grow very fast. These are like 35 days old plants on the right side. So the shade of the plants, the temperature according to the crop canopy, and all those things are studied very well before we actually jump into the field trials. Next slide, please. Okay.
It's coming, I think. Here we go.
So when we look at when we have the formulation, we have the blend, there are several parameters that we start looking at. So we make a list of several parameters that we need to understand the benefits of mating disruption. The first one is the moth captures. Okay, let me explain a little bit about moth captures. Why are we talking about trapping these moths when we're talking about releasing the pheromone into the cloud and creating a cloud of pheromone? So just imagine when you have dispensers out there in the field, several dispensers standing out in the field of 1 hectare plot through the season, they're releasing the pheromone. So we want to put a proxy female in the field, in the middle of the field, and then this proxy female is nothing but the pheromone lure. So the pheromone lure is nothing but acting as a proxy female. So if the whole area is saturated with pheromone source, the proxy female, which is nothing but the trap, should not be found by the males. So just imagine if we have a trap sitting in the middle and then covered with dispensers, the cloud of pheromone source will camouflage even the proxy female. If you don't, then you see the picture on the left side where you will catch more number of moths in no dispenser plots. So the whole understanding is we want what we call as trap shutdown. That has nothing but we want to have no moths, very negligible numbers in the pheromone dispenser installed plots or mating disruption plots. So this is the first thing, what we call as mode of action, right? So the mode of action is nothing but we want to have a cloud of pheromone. Is it enough with 20 dispensers or 30 dispensers? Those are the things that we actually look at several parameters as moth captures. And also it is important also to understand, we try to understand number of traps that are required to understand the efficacy of mating disruption. Okay, so ultimately what we want to understand is 0 moth captures in the mating disruption plots. Next slide, please. The next few parameters are very general parameters, just like any chemical insecticide evaluation or a biological insecticide evaluation. So those are the things that we start looking at other than moth capture parameters. Okay. I think it's coming through. Okay. So when we start looking at the moth captures, we also want to look at mating tables. So just imagine these females are waiting for the males to find them and then copulate, right? So we really want to understand if the females are mated or not inside the mating disruption plots. So what we do is we use mating tables, and we actually use sometimes we use sweep nets to recapture these females from the field. And it is not an easy task because most of them are nocturnal moths, right? So including the fall armyworm. So we need to go a little late in the night or early in the morning to recapture these females and dissect them to understand whether they have spermatophores or not. Spermatophores are nothing but egg sacs. So whether they have mated or not, that's the way to find it. So we seldom use light traps also, but light traps will attract not just the fall army or moths, but it attracts all kinds of insects. So it's a chaos over there. So we try to use mating tables and or sweep nets to capture these females, dissect them to understand if they have developed spermatophores or not, trying to understand if they have mated or not. Okay. So once we do all these aspects, then we look for egg loading and larval population. So the female moths, after the copulation, they go around and lay eggs, as we see. It's laid in egg masses. So we usually go around, collect the parameters of egg masses in the field, and also look at the larval population, just like any larval population that we look at for chemical insecticide evaluations. So the egg masses, though they have laid, we have seen several times that these don't hatch fully because either the female is not mated, they have to produce eggs, they lay eggs, but these are non-viable eggs. So they don't usually hatch and produce larvae. So that's a benefit of mating disruption. And we also want to understand the long-term benefits of mating disruption, where we look at the decrease in fecundity that decreases the number of generations season after season. Just imagine if there is a lesser mating happening in the field, there is a significant drop in the larval population. So very significant levels of drop in pupal population, and the moths coming for the next generation would be lower and lower. So with the regional mating disruption, season after season, we can probably see very negligible pest incidents, and sometimes we can even eradicate the pest in these situations. So these are all the several parameters, and the other parameters include damage reduction. We need to talk in the same language as the Davis scale. We use a Davis scale of 0 to 9, 0 being no leaf damage at all, and 9 being the whole whorl or fur leaves mostly destroyed. So we go and look at the damage reduction due to mating disruption. Lesser the incidence of pest, less is the damage. So it should directly correlate between mating disruption or lesser number of moths mating and the damage reduction that we see in the fields. So mating disruption is strongly correlated to the population density. So we have seen that in many cases where there is outbreak of pest population, it is very difficult to do the mating disruption, to rely on mating disruption alone, than compared to unifying other technologies, including chemical applications. In low to medium pest population densities, we have seen that mating disruption alone can do a good job. So we look at the damage reductions. These are all the parameters that we'll start looking at when we start doing the field trials. When we start doing the field trials, what do we compare with? Right. So if there is an insecticide evaluation or a biopesticide evaluation, we can easily compare with synthetic chemistries or conventional insecticide chemistries or conventional methods of management. But mating disruption is completely different technology. Mating disruption is a prophylactic application to prevent the pest population right from the beginning. So just imagine these dispensers are put out in the field right after sowing, and then these dispensers are taken out during the harvest season. So they are out there in the field protecting the crop throughout the season. So it is completely different compared to insecticides, which are typically used as a curative application. So you see the larvae, or the farmers see the larvae, they want to kill the larvae, and then they destroy it. So there is no direct comparison that will always make sense. However, we need to compare that. So we start comparing our mating disruption plots with synthetic chemistries or conventional grower practices to make, though there is orange to apple comparisons, but we need to make a comparison because there is no other technology that we can compare it with. So this is part of integration of several tools as a package for IPM. We always talk about integrated pest management, but at the growers level, they have very few choices. So probably mating disruption can be a choice to start looking at integrated pest management as a toolkit to manage fall armyworm and other pests. And then what we do is we start looking at the first few trials, what we call them as dose rate optimization. Dose rate optimization is nothing but, if we are looking at a biological insecticide or a synthetic insecticide, we talk about dose rates as number of or ml per hectare or gram AI per hectare. So the similar way we talk about dispensers per hectare. So we start looking at multiple dose rates in replicated trials to understand the dose rates for reduction in moth capture. So these are done in small size trials. When I say small size trials for mating disruption, it can go up to 30 hectares. As the below figure indicates, these are different locations. Just imagine I'm testing 5 treatments and in 5 replicate plots. So the whole area of location 1 is a 30 hectare plot. So why we do in 30 hectare plot is these pheromones are released out into the air and they're dispersed because of the wind. So smaller plots, smaller than a few hectares, will not be good enough for us to understand the benefits of mating disruption. So we need to have a minimum hectareage to understand the benefits of mating disruption. So once we do these moth capture reduction in the dose rate optimization trials, then we go for wide area trials. In the wide area trials, we choose commercially important areas, which are commercially important areas when I say are the definition where corn is grown in a larger hectare season after season, and where fall armyworm incidence is seen, so that we can have a higher probability of success for the field trials. For example, when we started doing trials in Indonesia, we choose East Java as one of the important locations where they grow corn at 31% compared to other locations in Indonesia. So we go there and then we choose the plots, we work with the farmers, we choose plots, and then get into an agreement with the farmers to do these wide area trials. So when I talk about wide area trials, these are like, 2 to 3 dose rate of trials replicated at least 3 to 4 times for their statistical significance. So a single trial location can have at least up to 60 to 70 hectares. So when I say 60 to 70 hectares, it is easier to do this in geographies like Brazil or Mexico, where corn is grown in a larger hectares, where the land holding of the farmer itself is 100 to 300 hectares. But when we talk about Asia Pacific region, the land holding of the farmer itself is less than 0.5 hectare. So we need to get into farmers agreements with so many different farmers, let them know what this technology is all about, and help them understand that this is going to show a lot of benefits for managing fall armyworm. So we also understand at the same time, we need to understand how many number of hectares is the minimum plot size that is necessary to realize the benefits from mating disruption. So these wide area trials are done at several locations for gaining initial understanding of the dose rates before we start the registration trials for registering the product. So when we talk about registration, it also talks about the viable product concept for commercialization purposes. So once these wide area trials are done, we have a lot of data with us. We need to churn the data. We need to understand what data is telling us. So when we talk about the data interpretation, so there are several things that come into picture. These are really large hectare plots, not a small hectare plots. So the statistical validation of mating disruption for decision making would be very, very important. So since these are really large hectares in several geographies, and sometimes we want to interpret the data from country to country, region to region, then we need to understand, then we cannot use the normal traditional statistics to understand the data. So what we use is spatio-temporal statistical analysis, where we come up with statistical models that are developed indigenously with us for understanding the space and time and the growth stages of the corn plant as well. So we use something called covariance matrix. This is all, sorry for the busy slide. So we use Gaussian process and probability success and general linear regressions to interpret the answers by tagging each dispenser for GPS coordinates. Why we do that is, for every dispenser, there is a pheromone cloud. The dispersion of the pheromone cloud through space and time is very, very important for us to understand. And not just that, the surrounding areas in that region also has corn. And we know that fall armyworm can fly to a longer distances. So it's very, very important for us to understand spatio-temporal analysis to understand the data and interpret the data, so that we understand what these benefits are really coming from. So traditional statistics do not really support us, so we use the spatial analysis for this. Once we get this spatial analysis done, then we start looking at the value creation trials for the growers. These are nothing but knowledge sharing sessions. Since the technology is very, very new, we need to make sure that growers understand what mating disruption technology actually provides them. So there are several ways of looking at the value and the benefits for the growers. We tell, we educate them in terms of proactive way of pest management, reducing and preventing the population growth in wide areas, reduces the residues, because we are the lesser number of generations, the lesser the damage it is, the lesser number of insecticides that need to be applied. So there are several benefits coming out of that itself. Residues are lower, the pollution and the health hazards are lower. We increase the natural enemies in the whole ecological system, and this is non-lethal to higher mammals and non-target species. So a single treatment can be effective throughout the season. You don't have to go and change, so less laborious. So, and then consistent area-wide utilizations. Farmers can come together and lessen the population in the whole area as a community farming. So we usually do these as value creation trials, show the value to the farmers because it is new technology. And then after that, we go for pre-commercialization phase. Call it as a commercialization phase, but it is more of a network and stakeholders involvement for new technology and knowledge sharing sessions. Because seeing is believing. This is a new technology. They need to see and believe and we want to showcase the technology and demonstrate value to the growers so that they can adopt this technology. At the same time, we look at the viability of the concept, whether it can support commercialization or not. So the pictures that I'm showing here is from the rice product that we're going to launch in Indonesia for yellow stem borer management this October season. So when after the pre-commercialization phase, it goes to the farmers, and then they start looking at adopting this technology as a tool of integrated pest management. This is my last slide. Briefly, I would like to talk about the constraints of mating disruption. So as I was mentioning to you, there are several farmers who are involved in this, small holding farmers. It's always a challenge to work with individual farmers. For each trial, if it is a 60-hectare plot, we're talking about more than 200 farmers who are involved in these trials. So it requires a lot of synchronous community-wide adoption of this technology. So it gives us an opportunity to work with the farmer groups, associations. It'll be more power for the farmers to manage invasive pests like fall armyworm more consistently. The second is fall armyworm is a new pest for Asia. So farmers usually use more than required insecticides, number of applications. They're scared. So it gives an opportunity to educate the farmers and then initiate a dialogue of economic threshold levels. So as an IPM practice. Finally, there are no guidelines as of now for registering mating disruption products. Though the pheromones are regarded as very safe, still we actually use insecticide guidelines. So it's an opportunity for us to network with our regulatory bodies in several countries for this adoption of new technology and speed up the registration process and implementation, of course, to help the farmers. Okay, that's where I end, and I guess I will pass on to Roma.
It'd probably be me, but Roman's going to come on very soon. But thank you so much, Pathi. That was extremely interesting, and you covered a lot of points in that presentation as well. We actually have a lot of questions coming in, so it's good we've got some time now. We've got 15 minutes actually, so that'll give us plenty of time to ask all of those. Just one thing that you mentioned at the end about guidelines for pheromone products is for the regulatory process. Is that something that you and other companies could work on together, or do you think that would be useful to have guidelines specifically for this product?
Usually mating disruption is with the pheromones that are used in traps. But these are these has to be tested for their efficacy. And when we talk about efficacy, we talk in a similar lines of insecticides, which is not that, which is a completely different technology. So I think we need to have a different guidelines. How do we really evaluate the efficacy of mating disruption products? That's where our protocol discussions and guidelines would come in.
Okay, excellent. Okay, moving on to some of these questions. I have one here at the very start is, can we import, I think, more of this technology from Provivi to the Philippines? If yes, how? Are you looking at other areas outside Indonesia at the moment in this region, or are you sort of focused more on proving it in one place before moving to others?
Right now, we are doing research in several countries in ASEAN, including Indonesia and Thailand. Yeah, we can definitely take Philippines into consideration as well.
Okay. I have a specific sort of more technical question here is, please advise the best method to conserve females without damaging their pheromone gland. Would you be able to answer that question?
It doesn't affect the pheromone glands of. I did not quite get that question. So, what's that question again?
No, I'm not sure as well, and I've just got rid of it. But it was more around, can you? I'll come back. Here we go. Wait a minute. I'm going to go. Please advise the best method to conserve females without damaging their pheromone gland.
It doesn't affect any pheromone gland of natural females.
Okay, there we go. Quick answer. Why do we not focus on treating host plants with semiochemicals rather than placing the dispensers in the field? Because is the air, it's quite difficult to maintain the concentration of, I guess, the pheromone, is it?
Yeah, yeah.
So I guess they're asking why, yep, go.
I think I understand that question. So
Yep.
the pheromone dispensers are non-crop applications. These are nothing but off-crop label usage. So we're not spraying anything on the crop. So when we spray something on the crop, it'll be much more be effective, but at the same time, we need to have multiple applications. So we need to also look at the economics of the farmers to spray these sprayable pheromones. For example, in Brazil and Argentina, we are actually working with sprayable formulations of pheromones, where we actually spray them aerially. But when it comes to Asia Pacific region smallholding farmers, I think it would be much more economical to use dispenser technology.
Oh, that's an interesting, that's a good point. And actually on that, you talked about the work that you do with farmers, which I think was extremely interesting. When you get farmers together to talk about this, to educate them about the technology, what are some of the sort of maybe one or two questions that spring to mind? Are there some common ones that come up time and time again that you hear from farmers? What's their first reaction?
Yeah, the first question that we actually get from the farmers is, okay, so we don't have to spray insecticides, right?
Okay.
So that's the first question. So yeah, there are many times what we have seen is we completely eliminate the number of applications of insecticides, but not all times. So insecticides are part of integrated pest management program. So we're not trying to eliminate insecticides, but we are trying to conserve these insecticide molecules for a longer time. So there are very few insecticide chemistries that we have. I think it is very important both for insecticide chemistries as well as traits for us to conserve for a long time usage. So this technology will actually help that lengthening the process of resistance monitoring.
Oh, good point. Excellent. Thank you. Here's a question from Audrey. Hi, fall armyworm presents a high fecundity rate, which means that you could have important population outbreaks. My question is how mating disruption is working for this species if you already have a large number of females present in the field. Even if the males will be perturbed, they will still find females. How is mating disruption then efficient against fall armyworm?
That's a great point. So yeah, for fall armyworm, especially compared to other noctuid moths, it is a very strong flyer. So we do these when we're doing these trials. Just imagine this is a small little island of mating disruption trial in a big sea of corn growing all around. But when there is an adoption of regional adoption or a village adoption or sub-district adoption of mating disruption product, what we have seen is there are very less number of mated females flying into the region So we're not disturbing females. So female moths are very welcome to come and lay the eggs in the mating disruption plot, right? So we often see the female moths which are mated outside the mating disruption plot, flying into the mating disruption plot and start laying eggs where we see the damage. But when we have an area-wide adoption, there will be very less number of mated females that we can actually see.
Okay.
So yeah, we need to have an intervention with some of the biological insecticides pre-applications, even in the mating disruption plots, especially for fall armyworm management, because it is a very strong flyer and it migrates from like southern part of US to all the way to Canada. So we can imagine that it is a very strong flyer. And we have seen that in Asia Pacific region also. Within a short period of time, it went all the way from India to Australia.
Yes, yes, indeed. And onwards into the Pacific as well now. In the case of fall armyworm, how many traps is recommended per hectare or area of maize for effective mating disruption?
It can range from 30 to 40 dispensers per hectare, but at the same time, we're still finalizing those dose rates for Asia Pacific region. We have launched the product in Mexico by Firogen. We recommend 30 dispensers per hectare, one-time application, which is one-time installation in a season when actually they do the sowing of corn plants or sowing of corn seeds.
Okay.
It'll stay throughout the season.
Okay. And when is the best time to install the dispensers? If the area site is often windy, does that have an influence on the effectiveness of the treatment?
It doesn't affect the effectiveness of the treatment with temperatures, relative humidity, and wind velocity and rainfall. The dispenser technology is helping us there. So it releases a pheromone cloud, or it releases a specific amount of pheromone through the day, and that will last long for season.
Okay.
Yeah, so it is the dispensers are placed in the field right at sowing. Just imagine as soon as the farmers are sowing their corn, either by mechanical means or by manual means, they go on the same day. A person walking behind will actually put the dispensers for every 22 feet or 22 pace lengths,
Okay.
so that these are evenly spread throughout the plot.
Okay. A question here. It says that pheromones are species. It is said that pheromones are species specific. Why are some catching other species? Do you find that?
No, we don't usually find that.
Okay.
There are some pheromones that are within the species as well, but that is very, very rare. For fall armyworm, if we are designing a sex pheromone source for fall armyworm, it attracts only fall armyworm, nothing else.
Okay. You've got lots of questions here, Pathi. I just hope that you're ready afterwards because there's a lot of interest. Just carrying on with some of these questions. Here's a question here from an anonymous attendee, but the question is, could an improper setup of pheromones result in more pest instead of less pest?
No, this is not attracting. This is one of the frequently asked questions by the farmers. When we think about pheromones, these are attractants, right? So they think that these pheromones are attracting the pests from everywhere, from the adjacent fields or neighbors' plots. Oh, when I put these dispensers, probably I'm attracting more males, and the females will find them. No,
Yeah.
that's not the case. This technology would prevent the mating wherever we have this pheromone cloud in the field. So it does not have an effect on attraction or bringing the pest into the plots.
Okay. And should the mating disruptor installation does it depend on the flying zone or the target pest, or is it at a particular height that it needs to be at in the plant canopy, or?
Yeah, it all depends on the cultural practices. For example, where we have mechanization or intercultural practices, where the farmers use small tractors, where we put the dispensers very close to the ground so that it doesn't have an effect on the intercultivation practices. But it
That's a good
doesn't.
point.
It doesn't. For example, we have seen that the dispenser technology will be fine if it is even put at 2 feet high.
Okay, excellent. What is the minimum field plot size to evaluate pheromone lure in the field?
That's a great question. So
That's an anonymous person, so.
that is a very good question, and it's a very important question as well. That's one big question that we're trying to answer in Asia Pacific region, where we have small land holding of the farmers. But in areas where, for example, in Indonesia, farmers work as a kopoktan or kolompoktani. It's like farmers groups.
Mhm.
So farmers plant corn in communities. So it is important. It will be easy for us if there are some farmer groups, associations who actually do collective farming.
Yep.
So where minimum plot size does not really matter at all. But minimum plot size for this one would range anywhere between 5 to 10 hectares.
Okay, excellent. Here's another question about the effectiveness of it. But does the pheromone lure lose its effectiveness rapidly in hot weather?
Okay, the first important thing is this is not a lure, it's a dispenser.
Yeah.
And the dispenser technology compared to the lure is just imagine you have active ingredient in hexane, and then the lures are dipped in, or rubber septa, I would say, are dipped into the active ingredient, and then they're placed out in the field in the traps. So it is pretty much exposed to most of the weather parameters. In the dispenser technology, the active ingredient is inside the dispenser. The dispenser is protecting it and releasing it through the season.
Yeah.
So the effectiveness is season long.
Okay. Here's a question here. Now, thank you for your very nice presentation. It starts, and then its question is, I realize mating disruption and pheromones way of working is different from biopesticides or pesticides, and efficacy is calculated differently. Still, should there be an average percentage efficacy against fall armyworm, and how much would you perceive to be the efficacy range?
Great point. Yeah, so it is different from biological insecticides. This is not an insecticide. This a mating disruption is a completely semiochemical management or sexual confusion. So when we look at the efficacy part coming from only from the mating disruption, irrespective of insecticides, or when we don't spray any insecticide or intervene, we have seen anywhere between 40 to 60% effectiveness.
Okay. Excellent. There we go. Good answer. Thank you very much. Here's a question here, and I think then we might leave it because that's the end of our session. But this question is based on your observation, how long does it take for the mating disruption strategies to show a decline in moth population in the field?
We still have to see that for fall armyworm as an invasive pest in Asia. But what we have seen for stem borer populations is we have seen that at least 3 to 4 seasons, so back-to-back seasons, what we call as year-round mating disruption. So if the same area is treated with mating disruption season after season, we see that the number of generations coming down drastically. And also for each generation is not that huge, so it'll be easier for the farmers to manage it properly.
Okay, great. Pathi, thank you so much for the presentation and all those questions. And if you could go into the Q&A box afterwards and have a look, I think there's a few in the chat. If you've asked a question in the chat, please put it in the Q&A, just a reminder to everyone. But you could, if you could go in there, you can actually write some answers if you've got time, Pathi, and that would be most appreciated. Extremely good presentation. And just before you go, I wanted just to give the floor to Roma. Roma, do you have any question that you would like to ask? She's on mute. Oh, she can't talk? And I may not have got her. No, I'm still muted. Says host is stopping me. I'm not stopping you, but let me see if I can ask to unmute. Does that work?
Yes, thank you.
Oh my goodness. There we go. I just
Sorry, I've been
sure Roma
unmuted
was
for the
having a
last
slight heart attack.
45 minutes.
No, that's
So-
perfect, just in time. But sorry about that. But I was just going to ask you, did you have, because I know today that was an excellent presentation, lots of good information. And now we're moving on to this part 2 of the effective design of biopesticide trials. thinking about that case study that Pathi just presented there, was there anything specifically that you had to ask him that might be a nice entry into the next section, or would you like to get started?
No, I've got nothing specific I'd like to ask, but I'll just pick up on that and get started if that's all right.
That would be great. Thanks, Roma. And thank you, Pathi. Thank you so much for the presentation. Very interesting. And there's lots to unpick there for actually other sessions around pharma communications, the regulatory side, etc. A lot of things that we've heard before, but it's great to hear it again. So thank you very much. And for all of those who asked questions, there's still 23 questions, and I think there's probably 8 questions in the chat. Very popular topic. So well done, and thank you very much. So Roma, you're welcome to start.
Thank you, and thank you, Pathi, for your presentation. I think Pathi's given us a very good example of working with a substance, a pheromone that's acting as a mating disruptor, and just exactly how different the trials have to be. You know, he highlighted that you're working on a landscape scale. He also highlighted that you're working in population management. You're not talking about pest kill. And because of these characteristics, you have to have a very different trial design. And I think Pathi gave us a very good example of that. So what I'd like to do now, if my screen works, is move on to the next slide. I don't seem to have got control. Could somebody give me control back?
Seems to be. You've got some big problems today with the...
It's not me.
Who can start sharing? You should be able to have control right now. Let me see. Pathi, you don't have control by any chance, do you?
No, I don't.
Okay, I'm going to see if... I'm not sure why you don't have control, but let me have a look here and... No, but I'm going to have to take over, I think, and find the... I'm going to request remote control.
Okay, I've got it now.
Oh, have you? Well, it may go to me now.
Let me just... I think it's fine.
Oh, you've
now.
declined it. Excellent.
Yeah.
No, you've got it. All good.
Lovely.
All good, everyone.
So no one touch anything, please, and so we can get it working. I think what's happening is if somebody else touches their screen, it takes it away. So great. Lovely. Okay, so if we're just thinking now about biopesticides, and I'm thinking primarily here about microorganisms and botanicals, Pathi's given us a really good explanation of what you need to do for pheromones. So if we just focus on those. So thinking what we know about biopesticides, we know that the laboratory to field transfer is very poor. That what really you can do in the lab is you get a yes, no. You don't really get good enough quality data to make any other decisions than that. We also know that something I want to highlight is the reference treatment, your standard chemical that you have in your trials. Often for a biopesticide, they're not available because you're comparing against, you want to compare against another biopesticide to see, this is what I expect this biopesticide to do. How does my test product compare to that? But often we don't have already registered biopesticide that we can use for that, and therefore what we're using is a reference treatment that's a standard chemical. And what I often see happen then is that somebody said, oh, it's not as good as the chemical, and we think about the question that we're asking. I didn't actually ask if it's as good as a chemical. I asked what was if I put no treatment in, what would happen? So I think we need to think carefully about what the reference treatment is and how we use it. We also know from the previous session that dose finding is not simple, that we often don't know what our dose response curve is, and it's very difficult to find that out. We know that variance is high, and as Pathi has pointed out, that the effect of a biopesticide may be long term. It may be across a whole season, it may be across multiple years as well. So again, this changes how we design trials and what we can do with trials. And often applications of biopesticides, we tend to think of things as that standalone, but actually we want to use them within programs or a series or within an integrated pest management program. Again, that makes it complex for trials. And we'll go on, and I need to think about application methods, water volume. And something we also need to think about is if you're working with something that's very new, often the regulations require you to have crop destruct, and this can end up being very, very expensive if you're working on a large scale. And you can imagine if you're working with a mating disruption pheromone and you have to have crop destruct, it's not feasible. You economically can't do that. So what I want to do now is break down all the different steps of when you're looking at a trial, and sort of my aim in doing this is so that we can think about how we can reduce our variances, and so that we stand a good chance of being able to have a result at the end of the trial that gives us an indication about how our product's working. So the first part we're doing is, okay, what's the test product that I have? And what we need to think about is saying, okay, what's the type of formulation? Why do we need to think about that? Because we need to think about how that formulation is going to go in the spray tank. We need to think about that type of formulation in terms of how I expect my product to work. We need to think about the specification to targets. This project, we're mainly thinking about fall armyworm, but sometimes we're looking at, if we're looking at diseases, we may have an expectation that it'll affect 2 or 3 diseases at the same time. Or if you're looking at an aphid, will it affect several aphids or just one aphid? And what am I going to calculate? So you need to look quite closely at the label to understand that. And very importantly, we need to know what the mode of action is. So is this a contact acting product? Is it systemic? Is it repellent? Because this is critical to how you design your trial. You also need to know, is the product persistent? We need a repetition of the target. We also need to know what crops we expect it to work on. And we need to look at the labels and say, well, are there conditions under which I shouldn't use it? For example, if it says this product is sensitive to UV, then I need to think about, well, should I be setting up my trial in the morning or the evening? And clearly, if something's sensitive to UV and you can set up the trial in the evening, you have a window of time where you're not having such a strong effect with UV. And it's baculoviruses, in which there is a baculoviruses for fall armyworm, are particularly susceptible to being killed by UV and light. But other products have conditions that they require for a temperature and humidity. So again, you need to pay attention to that. And then also have a look at what the application dose and delivery requirements. Now, I know what I'm saying is sort of standard. We should do this for every trial. But what I know from working with microbials and botanicals is they're very unforgiving of making mistakes, because they're not highly toxic, because they're not highly systemic and highly persistent. If you don't get it right, it shows itself. So this is what makes sometimes biopesticide trials a bit difficult, is it shows how bad we are at doing trials sometimes. So the starting point I have, having looked at my product, having looked at what they expect the product to do, and what actually the label says, if there is a label. The first thing I then do is think, okay, what is the question I'm actually asking for this trial? What am I trying to find out? And it's surprisingly, we're taught this when we first start to do trials, but sometimes we forget to do that and really think through is, what's the question I'm trying to answer with this trial? Because unless we understand that, we can't design the trial well. So are we asking, do we want to find out what the mode of action is? Do I want to find out what the dose response curve is? Because if I'm trying to find a dose response curve, I'm going to need multiple different rates of my product to understand that. And how many different rates do I need? Do I need 3, 5, 7? How many different doses do I need? I also need to understand, am I looking at the interaction of this product with the ecosystem? So am I looking at the effect of UV? Am I looking at the effect of humidity? And again, you're going to change your trial conditions if that's what you're asking it for. So perhaps if you've got a baculovirus, you may be doing some treatments which are applied in the morning and some applied in the evening, to say which one works better. More complicated is how to use something in an IPM program. That's really difficult to design trials. But doing that, it's much easier to work out how something works as a standalone. We also might want to try and understand how it interacts with a chemical pesticide. So if it's a microorganism, do I need to know whether certain chemical pesticides might actually kill or damage my microorganism? Or sometimes you've got something that you may have synergy with the chemistry, and so you actually want to think about how you use them together. You might not be able to tank mix, but maybe you can apply them in a particular spray sequence, which gets the benefit of both of the technologies. Also need to think about how long am I trying to find out how long the product persists once I've sprayed it. So for example, if it's botanical and the botanical is highly volatile, how quickly does it volatilize? Does it volatilize in 2 hours? Does it volatilize in 2 days? And maybe you want to find that out. And sometimes you might be wanting to find out the effect of an adjuvant with your test product. If I'm trying to find out how my new product is working, I always recommend don't put the adjuvant in, because the adjuvant, we already know how adjuvants work. We already know that adjuvants will usually add to efficacy. So putting the adjuvant in puts a layer of complication that's not needed. So I'd always recommend for biopesticides, when you're trying to understand how they work online, don't put an adjuvant in. But you might separately be trying to say, is adjuvant A better than adjuvant B than C? And see the trial. And then the other question you might be asking is, what's the best way to apply this product? So we forget sometimes that we need to go through all of these elements and think really carefully about what's the actual question I'm asking. And the way I sometimes try and think about this is, what do I want my label to say? Do I want my label to say apply every 7 days? And in which case, then my question I'm asking is what's the frequency of application? So I try and sort of think, if I've got the bottle in my hand and I've got a label, what do I want that label to say? And then I go back and say, okay, how do I design a trial to get that label information? So before I go further than this, are there any sort of burning questions that have come up that people would like me to address? Because this is a much more interactive session than last time, so really welcome your questions.
Not yet, I don't think, Roma, but I do see one here. Maybe it does relate, actually. In order to minimize interference amongst treatments or across treatments, what's the recommended separation distance between treatment plots? Now, this is probably to do with the last question, but I guess it goes back to your thing around what are you trying to achieve as well.
Yeah, actually, that's a really good question, and it is one that I'm going to address shortly.
Great.
So if I can hold that question, because
yes, you can.
That's actually very important and quite critical. Yeah. Okay.
Okay.
So the first part, so once I've worked out what the product's doing, what the product is, the next, and what the question that I'm asking, the next part is, how do I design my trial? And I've said just a short while ago, and just to say again, when you're working with biopesticides, they're very unforgiving. If you don't do things well, they show up, you get a bad trial. So we think about a trial design. So the first thing we think about is the treatments, what treatments are put in. So it's good practice to have a control, and you have your standard. Ideally a biological, but failing that, then it's going to be a chemical. The entire purpose of that is to understand how the trial worked. Then if you're testing the product and the question you're asking is, will this product have an effect on my target pest? I recommend to put in 3 rates of the test product, and you've got what you think is your label rate, then you do half your label rate and double your label rate. And what you're trying to see there is to understand how each of those rates work, and to understand how the pest is responding, and to also confirm that you're not putting more product out than you need. If you've got a biopesticide that has no adverse environmental or ecotox effects, or no adverse human tox effects, you might be tempted to think, well, actually, I can use it at a higher rate, but you've got the cost of it. So you're sort of trying to say, well, what's the best amount I can use relative to the cost? So as I think about what we're using the treatments for, so control to see, we want to see that there's no effect. What would happen if I didn't treat this pest? The standard, because I want to see if the trial design was good, and then the different rates, as I've explained, is you're looking to see if you could use less product, and also to see possibly if you have dose response. Now, I frequently see trials with more than 5 treatments in them. This becomes problematic when you have products with high variance, because you've got too many questions that have been asked, and usually what I find is none of them have been asked well. So I always recommend to sort of have a small tight trial design that asks just one question. Then you stand a chance of getting some good quality data out of it. So once I've worked out the treatments that I'm going to put into the trial, the next question is, what's the timing of those treatments? Do I put something in before the pest arrives? Again, chemicals are an example of this, or before the pest arrives, in the sense that, because often we detect the pest or disease long after it's actually arrived. So sometimes you think, well, actually, should I try and put something out there before I think I can see the pest? Or you might have got predictive models where you're expecting the pest to arrive and think, should I have an application placed out before it arrives? Now, if you haven't got a persistent product, that's probably not a good approach to do. So then we have put it after the pest arrives, but when after the pest arrives? Just after it arrives. And we see that in recommendations for trials is saying you should have 20% pest or disease. Now, for most of the biopesticides, 20% pest or disease is way too high. Your population has established too well, and it's very hard for the botanical, the microorganism, to keep up then, because what happens is you're starting to move into rapid population growth expansion, and it's that classic sort of ecological thing is your control agent is tracking behind your pest population, and what we don't want that to happen. We want our control agent to be in front of the pest population. So that question about after the pest arrives needs to be really thought about, and also to think really well about what the pest population is doing, how fast it's growing. And as I mentioned earlier, it's not necessarily about the size of the population, it's about how fast it's growing. You need to sort of think a little bit about that. So if we're looking at an application before the pest arrives, this is something, if you're stimulating plant defense mechanisms, this could be a good strategy. So you switched on your plant defense mechanisms before the insect turns up, and you're thinking, well, okay, so how long is that enduring? If that endures for 5 or 6 days, then I probably want to go in a few days in advance and apply it. After the pest arrives, this is really important for contact acting products, because if you have something like an insect pathogen like a Bavaria and it needs to actually be on the insect, then you need the insect to be there and moving around to actually get a dose. If you've got something like a baculovirus or a Bacillus thuringiensis where you're waiting for the insect to eat it, you can actually put the product out a little bit before the pest arrives, because the insect will move around and eat it and consume it. But so this is where you have to know your mode of action to know the best strategy. And sometimes you actually do want to put a product out when the population is growing fast. So if you think about something that's like a hyperparasitic fungi, where it needs the disease to be there so that it can actually germinate and grow. So again, this comes back down to understanding your mode of action well, to understand what timing you should use. And then we have to think about the frequency of application. Now, in an ideal world, we'd put the product out every day, because that's going to be the best chance for it to work. But we don't do that. Do we put it out every 3 days? Should we put it out at 7 days, or every month? What should we be doing, and why? Well, every day is just way too expensive and it's too much work, and no one wants to have to do that. And I know everybody loves every 7 days, because it means you can set a nice project plan and you can go out every 7 days, and the same thing happens every Monday. You go out to your field and you spray. But actually, if the population is growing really fast, the insects don't know that you like working on a Mondays. The insects are just growing. And so that maybe when your population is growing fast, you need to shorten your interval. So it might be thinking about that you have blocks. So say you have 3 applications at 3-day intervals. Then if you can see that the population growth is reducing, then you can open out your intervals to every 7 days or even every month. So again, this is where the frequency of application is not just depending on the mode of action of your substance, but also depends on what the insect is doing and where it is and what's happening to it. Something I just want to emphasize again is I don't recommend to be using an adjuvant unless you're specifically testing that. sort of can confuse your results and leave you in a position where you've kind of asked two questions together and you've not been able to answer either question well, because you don't really know how your product performed with the adjuvant. And I know sometimes people say, well, look at that treatment list, or I could double that treatment list. Each of the test products could be with or without the adjuvant. And yes, you could do that, but you then end up with a trial which has got eight treatments. It's a big plot. It's harder to assess. It takes longer to treat. And what I find is when you have a big trial with lots of treatments, people are less good at making assessments. They're less good at making applications, and there's more probability of things not working well. So again, I recommend to keep it as a nice tight trial with one question and just ask, have sort of around about 5 or 6 treatments. Okay, so has any burning questions come up, Alison, if we want to take
Yeah, there has been a few. And here's a very simplistic question here. How big does the plot size have to be? How do you work that out as part of your trial design?
Yeah, I'll come on to talk about that a little bit as well, but
Okay.
I'll answer it here because that is a really good question. So what you see, that depends on the pest, and it depends on the crop you're working on as well.
Okay.
So if you were working on something like an apple tree, your plot is, and you probably need sort of say 5 or 10 apple trees, your plots are going to be really big. But if you're working on maize and you're sort of thinking, okay, how many plants do I need to be testing? I would never have less than 10 plants. And you've got your guard rows, so you're probably looking at about 50 or 20 plants that you want to have in there. But you can do some mini plot work if you're asking a specific question. So the plot size depends on your crop, essentially.
Okay. Oh, great, thank you. I've got 2 questions here. Now I'm going to ask you both at the same time, both of them, and then you can decide which order you would like to answer them. One is, could you please elaborate a bit on how to test adjuvants? And the next one is, this is a trial design for applying on leaf. What would the trial design considerations be for seed treatment products?
Yeah, okay. So dealing with the adjuvants first, if I was doing a trial for adjuvants, I would have one rate of the product, the test product, and I would have it in combination with a series of different adjuvants. And I probably would only test 3 or 4 at any one time. So I'd have my control in there, I'd have my standard chemical with an adjuvant, and then I would also then have the test product with 2 or 3 different adjuvants that I want to test. That's the way I do it. So you wouldn't have a variable product rate as well as an adjuvant.
Okay.
And then I'm talking for seed treatment. For seed treatments then, so for seed treatments, you would be putting a different, if you're testing your test product and you want to know how efficacious it is, you would put a different concentration rate of material onto the seed. You'd stick it onto the seed at a different rate to get the same effect. So you haven't got the more complicated application step, but the principle of the trial would be the same.
Okay. Here's a question here. Can we use, I'm not sure, there's a word missing, but maybe are there challenges with using these products or testing these products with temperatures more than 30 degrees in high temperature areas?
Really good question, yes, and I will come on to talking about that actually. Yeah.
Oh, excellent, thank you. And there's, we'll come back to the high variance within and between treatments maybe later.
Yeah.
And I have another one here. Can we consider neighbouring plants as a factor in the trial and the application of biopesticides? Like, for example, nearby flowering plants, which is a habitat of parasitoids if the trial is pure botanicals or microorganisms?
Yes, you do need to think about the context in which you're working. I don't think you have so many concerns about the natural enemies in the surrounding area, because what we'd expect is the natural enemies if you have a product that isn't affecting natural enemies, you'd expect the natural enemies to come across the whole trial. And so what you're seeing happening in your untreated would happen in your treated as well. So it's a good question. It's something to ask yourselves, But I think often you can sort of say, actually, it's not going to be a big influencer on the trial, but it's definitely something you should be thinking about.
Okay. Okay, Roma, we've got lots of questions coming in. So what I propose is if we move on to the next section, just so we keep in the time,
Okay.
that would be great.
Okay, so having worked out what our product is, what our treatments are, what the next question was saying, well, okay, how do I design this trial? So I've worked out this model on the basis of having a 5 treatment trial with 6 replicates. Now this is a really important point, is I never work with less than 6 replicates for botanicals and microbials. And I also noticed that Pathi also was talking about 5 or 6 replicates. And the reason for this is how you manage your variance. So I often hear people say, oh, I've got very variable data, so I'll do lots of trials. What you end up with is that scattergram I showed you earlier, is you just end up with lots of trials with high variance. You don't reduce your variance. So how do you reduce your variance? You reduce your variance by increasing your number of replicates. But there's a balance, because if you have too many replicates, you can stand a chance of sort of hitting a hot spot and you get outliers. If you have too few, you're not accounting for all the variability. So you have to find a sort of sweet spot between the two. And in my experience, I've found that 6 replicates is about right. And I've done some work with a statistician on the probability of having outliers if you go more than this. So sometimes you can go up to 8, and if you've got quite a nice homogeneous pest infestation, you could go up to 8. But if you're not confident of having a homogeneous pest infestation, you need to be careful. But I would never go below 6, because part of that is it's quite legitimate if you decide at the beginning of the trial and the reason you do a randomized block trial is you think, I might have an effect of sun coming in from one side, or rain coming in from one side, or the influence of the hedge, or the influence of the neighbouring crop. So that's why you design a trial as a randomized block trial. And so you're already saying, I expect it to be slightly different. So what you're trying to do then is to have enough replicates, and it does, I don't sort of say this is a good way to go, but what it does let you do is, if one of those blocks has something happen to it which is peculiar, so for example, if somebody left the irrigation pipe running in one of your lines of pots and it just got completely flooded, you've got the possibility to remove one of the blocks and just do a statistical analysis on the remaining blocks. And that's the whole point of having a randomized block design, is that you understand those outside influences that you can't manage, and then you can statistically allow for that in the design. Whereas if you've only got 4 and you have to knock one of the blocks out, you're down to just 3, and therefore your variances can be massively high. So I really strongly recommend, and I've been using 6 replicates for about 15 years now, and as a way to get more consistent trial results. And something we always do when we're looking about plot layout is we always revert to the randomized block design. And I'm just encourage you to think about that and saying, do you need to use randomized block design? Why are you choosing a randomized block design? Not just pick it off the shelf, really think about that. So for example, why couldn't you use a completely randomized design? Do you expect there to be influences around the field where you're locating your trial that don't allow you to do a completely randomized design? Because that should be your starting point, is a completely randomized design. You go to a randomized block when you don't think a completely randomized design can work. So again, just encourage you to think about what layout you're using and why. And then a layout that I often use, and it's kind of my favorite, is I like the Latin square design. The problem with the Latin square design is you need to have as many treatments as replicates. But one of the things that I do then is I still have my 5 treatments, but I double up my untreateds. And that's another trick and another way in which you can get better statistical resolution between your treateds and your untreateds, is you have double the number of your untreateds. It's a way of helping to manage the variance in the data when you're analysing it, so you can see how much variability you've got, because you've got a stronger data set for your untreated that you're comparing against. And the nice thing about Latin square, as you can see from this, is that every treatment occurs in every row and in every column. And so that you know, they also then know that every treatment has always been surrounded by every other treatment that you're using. So you're managing the way in which things can influence each other. So I wanted to explain it first, and now I have sort of a little bit of a poll. Just to touch that, saying, somebody asked this, what happens if my product also stimulates untreated plots? This is a problem. So what I discovered when I worked with some botanicals a few years ago is the botanical, one of the things, its mode of action was it was directly killing the insect, but what it also does is stimulate plant defense mechanisms. And this isn't unsurprising that botanicals would do this, because this is some of their role in their plants. And what I discovered was when I treated my treated plots, were signaling, the plants were then stimulated by my product, and then those plants signaled to my untreated plots and switched on the plant defense mechanisms in my untreated plots. And suddenly what happened is I didn't have a difference between my untreateds and my treateds. And it took me ages to work out what on earth was going on in the trials. And I solved it quite simply by putting up fleece between the trials to cut down the movement, the airflow of the volatile, so to minimize the plant-to-plant communication. And that didn't completely get rid of it, but it really helped and it reduced it. So I started to then get the differences between my untreateds and my treateds. Another thing, then the other part I've already touched on is about what if my target is not evenly distributed? And I think you need to think about that and say, well, if it's not evenly distributed, can I change that? So can I artificially inoculate? Problem with artificial inoculation is, one, you can sometimes have artificially high level of pest. The other problem you've got is you've then got a very synchronous population, whereas when we want to test the products, we may be wanting to test it against an asynchronous population to make sure that it's more representative. So you have to sort of think through what is going to be the influence of all of the product on the crop and on the target, and also how well my target's distributed. And the distribution of your target also helps you choose your trial design. So I've spoken a little bit about plot layout, and I'm just curious as to what people have used. And so perhaps if you kind of like to do this poll and just sort of say, what trial designs have you used in your experience? Because we know a lot of the audience is very well experienced in doing trials.
I've just put the poll on in progress, Roma, so hopefully people can see that. We've got some people voting now. So just everyone, I think you have four questions there.
Okay. Yep, so you've got to scroll down to make sure you get the last question, otherwise It doesn't let you submit.
But the good thing is the technology is working now, so this is a good ending. But we do, it's 5:34. We are finishing at 5:45, so I'm just going to raise that. So I'm going to wait just maybe 10 more seconds, and then I'm going to end the polling. So quickly get your vote in if you haven't. Let's see if there's some last people. Oh, quick! I'm going to get over 50%. Right, I'm going to end it now. And I'm going to share results. You should see there. Can you see that, Roma?
Yep, I can see it. And it's sort of what I expected, is that we all tend to think about randomized block designs and maybe completely randomized, but we don't test ourselves with other designs. So I'd just encourage you to think about other designs and think why you've chosen the trial design that you chose. Okay, thank you.
That's a great question. Thanks.
Right. Okay, good. So then the other part I want to think about is how we apply the product. So we need to consider the water volume we're going to use, droplet size. So we need to think about the plant part that we're treating, and we need to really think about what the sprayer we're using and the nozzle type. So something I know from doing some work with some microorganisms is if we reduce the water volume from 1,000 litres per hectare to 500 litres per hectare, we had a 37% increase in efficacy just by dropping the water volume. So I often see people are working at 1,000 litres per hectare. That's way too high. Just way too high. But I do see successful trials in crops where people are working down at 100 litres per hectare. So don't assume that you need a high volume of water for biopesticides. So just think again about what could be a good water volume, and to think about what you're trying to do. And one of my biggest frustrations is people saying we spray to run off. If you're spraying to run off, the products all end up in the soil. You've just wasted it all. So there's no point doing that. So what you should be doing is spraying to, before runoff. You shouldn't be having your droplets coalescing. You should be really thinking about it. So then think about your droplet size. We know already if they're too large, they bounce off the leaves. They're too small, they may evaporate quickly. So why is this important for biopesticides? So if they're too large, you could have an accumulation of your microorganisms, for example, in those particles, and they disappear onto the soil. They never reach the target that you want them to. If they're too small, for a botanical, which is a highly volatile botanical, they can evaporate too quickly. And so you need to find a sweet spot between, particularly for microorganisms, where every droplet contains a microorganism. So you can't have them droplets too big because, and you can't have them too small because otherwise there won't be anything in them. So you need to think about that and find out what's the sweet spot to get the right number of particles in the droplet, or for a botanical, that you've got the right amount of water to slow down perhaps the rate of volatilization, that they don't evaporate too quickly. And also some microorganisms need water around them to germinate, so you might actually want a slightly bigger droplet for those. But that doesn't mean you put on more water, it means you manage your droplet size. Need to think about the part of the plant that's treated, and I was asked this question. Design your application equipment well for your foliar spray, whether you're doing a root irrigation, drench or in furrow, or whether you're treating your seed. And something for microbials, and that I really noticed as well, is really think about what kind of spray you're using, because one of the things I often see is people using recirculation tank. They get really hot, and as the water volume decreases, they get hotter, and basically you'll kill your microorganisms potentially if you do that. So check what kind of tank you've got using. Just also think about what agitation you've got in there and how much it needs to be agitated, and please use cold water. I so often see people taking water from the tap and they haven't let it run for a bit, and the hose has been lying out and it's really hot. You could potentially just kill all your microorganisms. So big appeal here is calibrate application equipment properly for the botanical and the microbial. It's not the same calibration as it is for chemicals, so that needs to be done well. Always use cold, clean water. You need to also pay attention to your pH, because the pH is going to have a big influence on your microbials and your botanicals. And don't leave standing in the sun. When I was working for a company, the number of times I turned up on a grower site about somebody saying they've got problems, and what it turns out is the guy had been mixing the product, then went off for coffee and just let it sitting in the sun. That's going to damage your product as well. And please read the label about any precautions and any things that you should do. So really you've got to have good sprayers, well calibrated, the right sprayer for the right technology, the right application technique, and really pay attention to this. This is probably one of the biggest contributors to trials failing for botanicals and microbials. So based on the time, I'm going to move on. So then the next part of that is sort of saying, so I've done my trial, I've got my treatment stuff, I've put them out, I've sprayed them really well. Now I'm waiting to see what happens. What do I do? Well, the one we always do is count dead and live insects. It's really easy, but that's not the only measure we should be thinking about. And particularly if you're working with something that has repellency, you're not going to find your insects at all because they've all disappeared. They're not on your plots. So you think about how do you assess that? So one, you've had to think about careful trial design. The second part is how do you assess the fact that all your insects have disappeared off your plots? Also think about, we look at disease or insect damage, and we look at what's the change in the insect damage. So instead of trying to count a live insect, measure the damage that's occurred there's some really good charts about the percentage of leaf area that has been damaged. And you can do some really simple things. It's like take the leaves off the plots and just actually photocopy them, and there's some really nice software that then calculate the damaged area that you could use. And so that can be quite a quick way to do it. And also, what's complicated with microbials and botanicals as well is you trying to assess how your population development has changed and the rate of population. So how can you assess that? And you also need to think that you probably need a higher frequency of assessment when you work with microbials and botanicals than you might do with chemicals, because you're looking for much more subtle effects and much more subtle changes. So the absence of a target is a good starting point, but it's not the only measure. You may also want to look at improvements in plant health. So some of these products which stimulate plant defense mechanisms, you can see better plant health. And when I've done some work on wheat, I can remember that my green leaf area on wheat was just atrocious. There was no green leaf area, but I had a much better yield on my treated plots. So you need to be thinking about what is your assess, what does my pest do, what does my product do, and so what is the best thing to assess for to get a measure of what it's been doing. Okay, I'm just going to move on a bit now and start to sort of wind up. So I want to sort of summarize now here and sort of saying, so when you're working with microbials and botanicals, what you're actually doing is you're not trying to kill pests or disease, you're trying to manage the population. And because of that, it's much more complicated. We have to think about the biological characteristics of the product. We have to think about the relationship between the dose and effect, and we need to understand the mode of action. And some of the things I try and think about when I'm about how to get good efficacy is, as scientists, we always tend to think about what's the best the product can do. But actually, a farmer is probably more interested in when a product's not working. So maybe we should design the trials to say, does this product not work when I only use at a very low rate, or does it not work when I apply it in the mornings or something like that? And so again, this comes into the beginning of when you're thinking about trial design, is sort of thinking about not how to make it work best, but how do I understand when it doesn't work? Because we have high variance, and because we can't manage what farmers will do, we can't manage that he gets the best, but we can manage that he can not do things. We can give recommendations as don't do this, or it doesn't work when you do this. So sometimes you need to think about efficacy back to front a little bit. Make sure you've got appropriate trial design. Really, please keep the number of replicates higher. Think carefully about your timing of application relative to pest and disease. Think carefully about the frequency, and not just go because it's easier to go every Monday morning, and just think of what the pest's doing. Think about what effect you're trying to do, you're going to assess, and really think about your application strategies. So I was going to pause there, Alison, and stop so that we could have a good discussion.
Yeah, no, that's great. And thank you, Roma. I always think you're a bit like a magician, really, because you've, I know I can rely on you to always keep it to the time schedule, and it's actually right on 5:44 on my timing. So that really sort of brings it to the end, but now we're going to have a question and answer time for those people who wish to stay on. I know it'll be a bit longer, but we'll have a very informal question time like we did last time, and so please stay on for a bit longer if you would like. I'm sure it's going to be very interesting, and I've got lots of questions here, Roma, for you. And what I might do is I might just tie it up, and then like I did last time, and just sort of finish it for those people that do need to go, and then we'll have that question time again, if that's okay with you. If you could just move to the next slide, and the next one, and the next one, and the next one. I'm just going to say thank you very much to both our speakers today. It was absolutely fantastic. And we're going to have questions now for Roma, which is just a really quite an in-depth discussion and presentation there that she shared with us. But please join us on the community at the forum. It's actually, there's a circle around the blog there. You can visit that too. And Roma is actually featured in one of those blogs from her session last time. And also, I'm just going to remind you that our session, we'll be carrying on with this series in 2 weeks' time, and there'll be a session there on conservation biocontrol. So if we just go back 2 slides, Roma, and we'll go back to questions, and or maybe even your finishing slide was a good place, I think, to end with. I just want to thank everyone for joining us and thank the 2 speakers. And if you'd like to stay for now for questions, you're most welcome to. And take care for those who are leaving. So, Roma, questions for you, and there's quite a lot up here, and I'm not surprised. Some very good tips. Here, let's start from the bottom. What should be the border size or buffer between treatment plots?
Yeah, this depends on your product, is what you expect me to say. So if you've got something like a botanical that has got is highly volatile, you want to think carefully about having either crop barriers or other barriers between your plots. And then for the outside of the trial area, I usually have at least one row of the crop around it. It's not always feasible depending on the crop you're working on, but that's the ideal, because that helps to make sure that each plot had the same humidity and agronomic conditions. And also simple things is people bump into your trials. That's one of the biggest reasons I lose trials, is people walk over and bump into, particularly the farmers. So it's quite nice to have an extra border around it and that you clearly mark it up. And I was taught to have the same border around every plot, but sometimes I know that's not feasible because of the size and space you're working with. So ideally, a row of crop all the way around and between plots. But if you can't do that, what I try to do then is that I only sample from in the middle of the plot area. So you've effectively created guards by taking your samples within the middle part of your plots.
Okay, and I guess it's the same. I mean, how much minimum distance should be kept in between treatments during designing the trial layout with any biopesticides to avoid any cross impact between the treatments? That was another question.
Yeah, so same thing. You should have guards around them, and again, the number of guards you have, whether it's a double or a single row of guards, depends on the product. So, and it also depends on how good your spraying is as well, because what you're trying to stop is trying to stop your spray moving into the plots, and you're trying to stop other move. So if you put a seed treatment, clearly it's less important than if you've got a foliar spray. But yeah, you should have guards around all your plots if you possibly can do it, because it's very rare that someone who manages to stop right at the end of their plot. So just make sure you've got a bit of space. I don't like it when there's big walking spaces between plots. You must have sufficient walking space between the plots so you're not walking across your plots. But I don't like when it's too big, because then effectively each plot becomes isolated, whereas what you're trying to do is reproduce what's happening in the field where the crops are closer together. So you just have to find that delicate balance, that you've got enough space to walk and not damage your plots, but not so much that each plot is then completely isolated from around it.
Very good point. Here was one that was at the start. Very often the major problem is high variance within and between treatments, especially for the pest density. What is the best way to solve it?
Replicates. Yeah. So no, it's be clear about the question you ask, keep your treatments as low as possible, and apply your products really well. And then it's replicates. We can't get rid of the variance. What we can do is design trials that manage that and allow us to read the data from it. That's what we're trying to do, but we can't completely change the variables. We can make sure everything's applied as well as possible. It will always be variable. You will always have 40% variability. So replicates, and if you really think you've got something with a high variance, double your controls so that you can show the difference between your untreated and your treated.
Great tips, Roma. A question here from Wilma. What should be done just in case you are not able to finish applying the treatments because of rain? So other treatments will be washed out while others have no treatment at all.
You shouldn't have been treating your trial at all if you thought it was going to rain. That's the answer to that. If it's going to rain, don't apply your treatments. It's the wrong time to apply.
Okay, good answer, very clear. And I think we answer this question, I think, about what happens with these temperatures, these very high temperatures.
We didn't answer that one. So what was the question? Yeah. So that was asked, wasn't it? What happens at very high temperatures? Yeah. Again, you need to understand the product, understand what the limitations of the product are, whether it is influenced by temperature. But for the botanicals, which are highly volatile, I would tend to recommend that you apply those late in the evening, so that you've got time for the product to work. Whereas if it hits really high temperatures, what tends to happen is the droplets dry out very quickly and the product volatilizes before you can have your effect. So think about changing when you make your application to balance it with the weather conditions.
Okay, here's a good question here. Should yield be an important parameter in biopesticide trials, or all pesticide trials for that matter, considering that yield is a function of many factors?
Yeah, it's a very good question, a very tricky question, because we often know that we've got products that we consider working and sell, and farmers like them because they kill insects, but actually it doesn't make a difference on yield, or we can't detect the difference. So I think the answer to that is it depends on the product you're using, and it depends on what you're trying to do as to whether yield is useful. So if you have products which stimulate plant defense mechanisms, that's probably a situation where yield is probably going to be more useful to assess than other situations. So again, it's just thinking about what the product is, what it does, and what is the question I'm trying to answer by assessing yield. Something I do see though is not just yield, is quality. So you can see quite often some products, if I'm thinking about potatoes, if I've used a microorganism to treat potatoes, what I can see sometimes is that I'm not seeing a significantly different level of disease, but what I can see is I have more marketable crop. Yeah. And because you get some effect, which means that you've got all the potatoes the same size, which sells better, you get more money for that.
Oh, that's really important though.
Yeah,
I mean,
yeah,
for
exactly.
the farmer, it's
incredibly
incredibly important.
Yeah.
Here's a question here from, and it's how to minimize spray drift effects.
Yeah, right. Yeah, so just going back to the last one, saying so. Yep. What I was going to say was its marketability is perhaps more important than yield, to emphasize. How can you spray drift effects? Don't spray when it's windy is one of the things. So that's not the right spray conditions. So make sure you're using the right spray conditions. Think about your droplet size. Have you got too fine a droplet, and so that it's prone to drifting? Look at your height above your crop as well that you're spraying. You just think about what nozzles you're using and the spray droplet size you're using. And the main problem with the spray drift is that you're losing product off your treated area as much as anything. So you're not getting the treatment to where you think you are.
Okay, a couple more questions, 2 more questions perhaps, and then we might wind up. But there is lots here. I see Peter Chinwada has definitely got lots of good questions here. He must be working in this area. In biopesticide trials where there is no ideal reference product, should the decision on target product effectiveness be dependent on performance relative to untreated control, or a chemical product used in place of a true standard?
It should when you're using the biopesticide, it is always, and it's true for anything actually, it's comparison to if I didn't treat. I'm not asking the question, did my product work as well as the chemical? I'm asking the question, how does this product perform compared to not treating? The only reason I have a chemical standard or any standard in my trial is to prove that my trial design worked. So what I expect is my standard would give me the level of efficacy as I know and expect it should do. I'm not interested in what it does relative to my test product. That's not the question. So the question should always be, how did my product perform compared to untreated?
Okay, excellent. Here's maybe 2 more questions. There's so many. But what is the effect of naturally occurring pathogens on efficacy trials of biopesticides? Is it something to consider? In such cases, mortality in control and increased mortality in treated plots. I'm not quite sure that didn't quite make sense, but I think you understand it. Is it something to consider?
Yeah, and I think we've talked about that. So saying that you're not working in a sterile crop. There's other pests and diseases in there that are going to be interacting with what you're doing. So yes, you do need to think about them. So if I was doing assessments, I don't just assess for the insect that I'm looking for or the disease. I'm looking at what else is there. So that's, are the natural enemies coming in there? Because again, natural enemies could influence your effects. But I'm also looking at what other diseases there. So you should do a full inventory, not necessarily in the same detail as you're doing for your target, but you should have an assessment of what's going on in the crop for the other pathogens and other natural enemies, and they may or may not have an influence, but you should still have a good idea of what's there.
Okay, I've got one more question. I think that'll bring us to the hour, but it is, what is your opinion on application of microbiome products during night is it more effective compared during the day? And have you heard about this?
Yeah, I have heard about it, and I just do wonder how you manage to work in the dark. I think it's that sometimes it's a misinterpretation of what people are trying to do. So it could be because the humidities are higher, but I sort of think you should apply in the light, and that if you make an application, but make it late evening before it gets completely dark, and you'll have good humidity. It's the same for UV. You're going to get less exposure from the UV, but you have to also balance that against, if your insect's not active at night either, and you're looking to have quite a quick effect, you're not going to get the effect that you think you should have during the night time as well. So yeah, you just need to think it through.
Okay, so thank you so much for this whole session, Roma and Pathi. I'd have to say it would be great to actually be physically in a location, actually talking about this and workshopping it and actually going out in the field. And I think that's something that actually all of us in the region would enjoy. So maybe that's something we can look forward to next year. We'll see. We'll cross our fingers there because that would be really great to talk about this in person, I think, and we could talk about it for many hours. And this was just a really an amazing session. Lots of useful tips. It's great to hear and have shared the experience of Provivi in the field. All those good ideas and really just from your experience, Roma, to share that. It's very valuable to everyone working here, and you can see the positive feedback that we've had. It's absolutely brilliant. And I've just got one more poll actually for everyone, if you're still here, but I would like to say thank you again. And I'd also like to say take care, everyone, and we'll see you back at our next session. Pathi, thank you very much for your help and for answering all those questions. I can see you've been very busy in the background, and thank you again, Roma. And no problems about the few little problems we had. It is actually an extremely good event, and sometimes those things happen. We all know that. But fantastic information shared. So thank you very much, everyone. If you could answer the survey, that would be most appreciated.
Thank you.
Thank you, Pathi.
Thank you. Thanks,
you,
Alison,
Roma,
for giving us a chance to do this. Yeah.
Oh no, fantastic. Absolutely fantastic. So thank you. I'll just give people a bit of a chance to answer it.
So do we get immediate feedback?
I've given you immediate feedback. You're absolutely amazing, both of you. It's really great. Fantastic to share that information. I think we've got someone that's doing their doctorate, I think, or master's, and they were just saying that they're going to use lots of these tips to think about in their design trials. So you can't ask for better than that.
Good.
Great. Yeah.
Thank you very much. And I'm going to close the session now. So take care, everyone.
Great day.
And I will see you later. Goodbye.
Bye.